Hello, everyone. Today, I would like to talk to you about part of my PhD thesis. Now, before I start my talk, has this ever happened to you that a person that you just met comes up to you and asks you, "What is it you're doing? What's your PhD topic about?" And this happens to me a lot in Germany. And the word in German for fragmentation is pretty similar. Some similar in English too. And whenever I say, yeah, I'm working on, in fact, of habitat fragmentation in reptiles and amphibians. And the response I would get would be fermentation. You mean like snakes in alcohol? See, the thing is, a lot of people in the public don't really know what habitat fragmentation is, except for conservationists and biologists. So, what is actually habitat fragmentation? It is a landscape process scale involving both a loss of area and habitat, and also the fragmentation between these habitat patches. And this happens across time. Now, this can get quite tricky sometimes because you do get two different processes, such one being habitat loss, the loss of area, and the other habitat fragmentation per se. So while controlling for habitat loss, so you have the same area of habitat, but the patches of habitats are more isolated or fragmented between each other. Now, if whether this fragmentation per se has a weaker or no effect compared to habitat loss, we're not really sure yet, but most researchers tend to not distinguish between these two processes and tend to be inconsistent. So to avoid this inconsistency, in this study, we use habitat fragmentation to describe these two processes. So why is it so important to this study? It is because, as you may know, habitat loss and fragmentation are the main causes of biodiversity changes in our nowadays terrestrial ecosystem. And you can see that over the past 30 years, there's a net loss of forest area, but also the local richness as well, especially in North America and Europe. But more concerningly, in the neotropics, Afrotropics, and Southeast Asia, you get this a 30% net loss of local richness. And when you look at conservation studies that's been done, most of it are still being done in North American countries, European countries, and also Australia, and very, very few in Asian Africa. And when you compare to known species richness, well, throughout the years too, you get a disproportionate studies in developing countries and continents where you have more or higher biodiversity. So we want to know if this study bias has improved over the years, and if there is a bias in the geographic distributions of the species studied in habitat fragmentation compared to the known species. And the second question we ask is how are researchers conducting the study? So is there any significant relationship between the methods that you're using, the measurements of response variables that they are looking at, and the type of habitat fragmentation? So we predict that perhaps genetic techniques are used in population structure studies. Spatial models spatial layers are used in ecological niche modelings. Species richness studies are mainly in fragmentation studies. And we could see possibly these clusters of categories of papers. And we want to see if these clusters or how these clusters are distributed across the world. So whether they have a distinguished pattern or whether there are many overlaps or more homogeneous distribution. So what we did is we went to Web of Science to look for papers over the last 30 years on habitat fragmentation for amphibians and reptiles, and we found 1,600 papers. And by removing the duplicates, we got 1,400. And we didn't really screen through the abstracts, but instead we look at papers one by one. And we include the ones that are peer-reviewed, that has data at species level, and the ones that did not. Well, we also excluded reviews, paleospatial data, and the papers that didn't specify any habitat fragmentation. and we turned out to have 700 papers for selected studies. So in these selected studies, we extract the location of study sites, which focal group they are, and what species that the researchers are looking at. And we also carefully categorize each paper to the extent that what forms have that changed that the researchers are looking at, what sampling methods that they're using, and what sort of response variables of the animals that they're looking at. So what we found out was that there was not really improvement in bias that we've seen before. So USA or North American countries has the highest amount of publications, and also the lines here indicate this, collaboration networks. So you have European countries which have high collaboration networks, but here I want to show you that Brazil is one of the leading experts in amphibians, which is very interesting. But the same cannot be said for reptiles, where you have, of course, although USA still has the highest collaboration networks and intensity in public in research, but Australia is one of the leading research groups or countries in reptile research. Now, when we look at locations of study sites across the world, we still see the same or similar publication bias. But what I want to show you here is, if you look at the proportions of studied species compared to known species across the continents, you see that Europe has one of the highest species studied. So compared to what is being known, so 2% of, although 2% of the described species from the world, it comes from Europe. And so you get this disproportionately poorly studied continents such as South America and Africa. And this is due to the lack of publications. But my point being is that many species are being ignored. And why is that? Several reasons is that perhaps there is lack of publications, but also more and more new species are being found in the tropics in more high biodiversity areas. And also worms and caecilians, so cryptic species are found there. And at the same time, researchers tend to focus only on well-known taxonomic groups. And for the categories, we use hierarchical clustering to remove redundancy between the categories, but also try to find out which sort of clusters that they belong to, or if there's any clusters. And we did found 3 main clusters, which I'll explain later in the next slides. And for each cluster, we name it as a research agenda. So you have this different agendas that the researchers are focusing on. So the first one and the main one being individual species response. And this is the most common response variable. So species richness, presence, absence, and abundance that researchers tend to use to indicate the effect of that fragmentation. And this was associated with agricultural papers and also fragmentation studies. But what I want to point out here is what's interesting is that the researchers also use functional richness and comparison between specialists and generalists to look at the impact of fragmentation, because sometimes species alone is a poor indicator of the functional value of the species. And by using functional richness, we get more information and reduce the redundancy. So, sorry, just want to explain. So the associations are actually gotten from the graphs here. So when we look at it across the map, you see that there is a similar bias in publications for this cluster, but also compared to the other clusters or to the other agendas, which I'll show you later, South America and Africa have actually quite a number of studies in species richness, for example. There's some theories behind it that high biodiverse regions that could be explained by greater sampling efforts, especially in identifying different species, but also there could be no access to highly specific expertise and up-to-date methods in these poorly developed countries or regions. And that comes to our next topic or next agenda, which is the use of genetics methods and niche models. So genetic methods were used in population structure studies or connectivity between population, but also ecological niche models or spatial models. So over the past decades, there has been an increase in genetic analysis because of the availability of genetic markers and new technologies. So studying this genetic structure can, at population level, can offer a higher resolution of species response. But at the same time, the popularity or the availability of spatial layers are increasing, and this was in tune with the use of ecological niche models to study habitat fragmentation, and sometimes also more recently used in conjunction with genetics, so landscape genetics. And this has been proven to be highly effective in conservation management. And when we look across the map, what I want to tell you was that you can see that Asia and Africa have way less research agenda in population genetics. And what we find puzzling is we assume that China and Japan, with their advancement of genetic techniques, there would be more studies. But then what we show here is that none of them were concentrated or were focused on conservation. And the last but not least, researchers tend to focus on physiological and movement ecology. So they would use direct tracking methods, experimental methods, and this would be used to study fitness or ecological response or behavioral response and so on. And also sometimes interaction between species. So what happens is that a lot of times researchers tend to use or design dispersal experiments to study behavioral movement changes. But the problem is that some of them could be highly costly. So they cost a lot to do. And this was also not really seen in poorly developed continents. And when we look at the three different research agendas plotted in the map, we could see that there is actually not much of a pattern here, not much of a heterogeneous pattern. So a good thing about this is that we see that researchers are sharing their methods across the globe. So you have, especially in North America and also Europe. But the sad thing is the lack of papers in other continents sort of limit us to see whether there's a real pattern or not in this research agenda. So we were also limited by the synergetics effect of habitat fragmentation. So whether you have interaction between different habitat change, this was not accounted for. And this could also reflect the real ecological impact in the world. And we did not also look at whether there has been improved or decreased biotic responses. And this is because it has been done countless times and little or no general patterns has been found. But we did identify promising research fields and geographic areas that should be used as guidelines and starting points. But we also expect near or similar geographic thematic patterns in other species groups. And lastly, we highly recommend high resolution or new technologies in genetics and niche models for further future conservation research. I would like to thank my supervisors, Dennis and Anthony, and also AKG and International Biogeographic Society, who funded my participation in this conference. So thank you very much. Any questions are welcome, and you could email me anytime. Thanks.